Imagine your brain is uh, a highly secure, incredibly complex mansion.
Right. A fortress.
Exactly, a fortress. And the security guards patrolling the halls of this mansion are your microglia.
Which are the brain's resident immune cells.
Yeah, and for most of your life, they do this fantastic job. They protect the house by taking out the trash, clearing away misfolded proteins, just keeping everything pristine.
They're essential.
They are. But what happens if those security guards suddenly become chronically paranoid? Like, what if they start shooting up the house at the slightest shadow?
Causing way more damage to the walls and the furniture than any actual intruder ever could.
Exactly. Welcome to a new deep dive. Today, we're jumping straight into a massive, groundbreaking 2026 study from the journal Neuron that looks at exactly this problem.
It's a fascinating paper.
It really is. We are going to meticulously trace a microscopic chain of events, starting from a tiny chemical tag on your DNA, all the way to Alzheimer's disease and memory loss, and we're not leaving any detail behind.
It really is a profound shift in how we view the aging brain, For decades, the dominant paradigm in Alzheimer's research was focused almost entirely on the accumulation of those sticky proteins.
Right, the amyloid-beta plaques and tau tangles.
Exactly. We treated those plaques like the root cause because they were the most visible damage. But the new frontier is understanding chronic neuroinflammation.
Because the plaques aren't the whole story.
Not at all. Today, we're looking at the exact molecular master switch that makes those microglial security guards turn destructive.
That distinction is just so important, whether you're interested in neuroscience, longevity, or just, want to understand how your own brain's immune system operates. Mastering these concepts will completely change how you view aging.
Absolutely.
But, okay, let's unpack this. If these microglia are built to protect us, why do they suddenly change their behavior? Where's the switch that flips them from protectors to destroyers?
Well, to find that switch, we have to look deep inside the nucleus of the cell. We have to look at the epigenome. We need to talk about how genes are actually stored and read. Think of your DNA as this massive, continuous thread of information. I mean, there's about 6 feet of it in every single microscopic cell.
Which is crazy to think about, 6 feet in one cell.
Right. And to fit inside the nucleus, that thread has to be tightly wound around these protein spools called histones.
So I'm trying to picture this. If the DNA thread is tightly spooled up, the cell's reading machinery can't physically access the code.
Right. The gene is essentially turned off simply because it's buried. You can't read a closed book.
Got it. So epigenetics is just the process of modifying those spools to turn specific genes on or off without ever changing the actual DNA coding cell.
Spot on. And this brings us to the master switch identified in the Neuron paper. It's an epigenetic enzyme called KAT7.
KAT7.
Yeah, KAT7. Or sometimes it's called HPO1. It's a histone acetyltransferase. It actually sits in a complex with a scaffold protein called JDE2.
Okay, I like to think of KT7 as a microscopic librarian.
Yep.
But this isn't a librarian who just hands you a book. This librarian actively loosens the binding of a tightly sealed book so the cell can actually peel the pages apart and read them.
That is a very accurate way to visualize it.
Yeah.
On a chemical level, KAT7 has one specific job. It adds a chemical tag and acetyl group to a very specific location on that histone spool.
Okay, and what is that location?
In the literature, it's referred to as H3K14SPC.
Okay, I have to stop you there because terms like H3K14SC can sound like pure alphabet soup to, well, anyone who isn't a biochemist.
Fair enough.
Break that down for us. What does adding an acetyl group actually do physically? How does it loosen the binding of the book?
It comes down to basic magnets, really. DNA naturally has a negative electrical charge. The histone stools it wraps around typically have a positive charge.
So opposites attract?
Exactly. Opposites attract, which keeps the DNA wound incredibly tight. But when KT7 adds that acetyl group, the H3K14X market brings a negative charge with it.
Oh, I see.
Yeah, so suddenly you have a negative spool and negative DNA. They physically repel each other. The chromatin structure relaxes, bowing outward.
Which exposes that specific stretch of DNA so the cell can read it. That is fascinating. The cell literally uses magnetic repulsion to turn on a gene.
It's incredibly elegant. And the researchers found that this KAT7 librarian is highly active in Alzheimer's disease.
Like way more active than normal.
Massively elevated. They looked at human postmortem brains from Alzheimer's patients, and they also used a specialized genetic mouse model. It's called the 5xFAD mouse.
5xFAD? What makes that mouse special?
It has 5 specific familial Alzheimer's mutations. So It rapidly forms amyloid-beta plaques. And in both the human brains and these mice, they found huge amounts of KAT7 and specifically that H3K14 ACK mark inside the microglia.
Just the microglia.
Yeah, mostly just the microglia that were huddled right next to the plaques. The other brain cells, like the neurons, didn't show this massive change, only the immune guards.
Okay, so let's do a quick recap here, right around our 5-minute mark, just a few bullets so we're all on the same page.
Sounds good.
Bullet 1, microglia normally protect the brain, but over time they can become hyperinflammatory. Bullet 2, this paranoid state is driven by an epigenetic enzyme called KAT7.
Yep.
Bullet 3, KAT7 works by loosening the DNA, adding that H3K14 ACK tag, basically pushing the magnets apart.
Exactly right.
Which is wild and makes you wonder, if your cells had a volume knob controlled by epigenetics, what genes do you think your lifestyle is turning up too loud right now?
That's a great question to ask yourself.
But here's where it gets really interesting. Once that DNA book is forced open by KAT7, what does the page actually say? Like, what specific gene is being read?
The primary target gene that KAT7 exposes is one called CMPK2. And it produces an enzyme called, you guessed it, CMPK2.
BMPK2. Okay.
And this enzyme operates entirely within the mitochondria, the energy factories of the cell.
Let's carry our factory metaphor further then. If the mitochondria are the factory, I picture this CMPK2 enzyme as a rate-limiting supply chain manager.
That works perfectly.
Its whole job is to manufacture raw materials.
A very precise raw material, actually. CMPK2 produces the fundamental building blocks called DNTPS, required to synthesize new mitochondrial DNA.
Wait, I need to pause here. Mitochondria have their own separate DNA. I thought all our DNA was locked in the nucleus.
They do. And this is a crucial piece of the entire puzzle. Billions of years ago, mitochondria were actually independent bacteria.
Oh, wow.
Yeah, they were swallowed by our single-celled ancestors. They formed a symbiotic relationship, but they kept their own distinct circular bacterial DNA. Called mtDNA.
Okay, so KAT7 gets upregulated, which turns on the supply chain manager, CMPK2, which turns out a massive excess of building blocks for mitochondrial DNA.
Correct.
So suddenly, the mitochondria are in overdrive, copying their own circular DNA like crazy. But why does that cause the microglia to destroy the brain?
Because of the environment these cells are in. In an Alzheimer's brain, these microglia are constantly encountering toxic stress from the amyloid-beta plaques, specifically these oligomers.
So they're under attack.
Right. And this external stress physically damages the mitochondria. Now, because our supply chain manager forced these mitochondria to overproduce DNA, they're essentially overstuffed.
Like a balloon with too much air.
Exactly. When the stress causes them to rupture, they leak this massive excess of mitochondrial DNA directly into the cytosol, the main fluid body of the cell.
And I'm guessing mitochondrial DNA is absolutely not supposed to be floating freely in the cell fluid.
Never. And remember we just talked about mitochondria originating from ancient bacteria?
Yeah.
When your cell detects circular bacterial-like DNA floating freely in its internal fluid, it doesn't think, oh, my mitochondria are leaking.
It thinks it's an infection.
It thinks, I have just been invaded by a lethal pathogen.
Wow. So it pulls the chemical fire alarm.
Exactly.
Yeah.
It uses a very specific immune pathway to do this called cGAS-STING.
cGAS-STING. Okay, break that down for us.
So the leaked mtDNA acts as a damage-associated molecular pattern. The enzyme cGAS senses this misplaced DNA and produces a messenger called cGMP.
Think of that messenger as a flashing red strobe light spreading through the entire building.
Right. That flashing light activates a protein called STING. STING then phosphorylates or activates other proteins called TBK1 and IRF3.
Okay, that is a lot of acronyms. But the ultimate result of this whole domino effect.
The result is the activation of NF-kappa and the NLRP3 inflammasome. Basically, the microglia start pumping out highly toxic pro-inflammatory cytokines like IL-6 and IL-1-beta.
Which are biological weapons designed to kill invading bacteria.
Yes, but there are no bacteria. It's a false alarm.
And those biological weapons end up causing immense collateral damage to the surrounding brain tissue, including the neurons we need for memory.
It's tragic, really.
Let me just synthesize this because the causality is incredible. So let's do our next bullet recap here.
Go for it.
Bullet 1, the KAT7 librarian turns on CMPK2. Bullet 2, CMPK2 makes too many building blocks for mitochondrial DNA. Bullet 3, the mitochondria get stressed, burst, and leak this ancient bacterial-like DNA into the cell.
Perfectly said.
Bullet 4, this triggers the cGAS-STING fire alarm and massive neuroinflammation occurs. Have you ever experienced a false alarm in your life that caused far more collateral damage than the actual threat?
That is exactly what your brain cells are doing here. The immune system's absolute desperation to defend the brain becomes the very mechanism that destroys it.
It's mind-blowing. But mapping this incredible molecular domino effect in lab dishes is one thing. How do scientists actually prove this causes Alzheimer's symptoms in a living, breathing mammal?
You have to work backward. If you believe this specific KAT7 enzyme is the root cause of the paranoia, you have to remove it and see if the paranoia stops.
But you can't just open a mouse's brain and watch a microscopic mitochondrial leak.
No, you use genetics. The researchers used a highly advanced technique called easy CRISPR to create a conditional knockout mouse.
Okay, I have to push back here. If you genetically engineer a mouse to lack a specific gene, doesn't it lack that gene everywhere? Heart, liver, neurons. How do you isolate this mechanism only in the microglia?
That is the genius of the conditional knockout. They used a two-part genetic lock and key system called CX3CR1 Career.
Okay, help me visualize that.
They bred a mouse that had little genetic brackets placed around the KAT7 gene. Those brackets do nothing on their own. Then they introduced a second genetic sequence, a microscopic pair of scissors called Cre recombinase.
But the scissors only cut the DNA between the brackets.
Exactly.
Yeah.
And they engineered it so that the scissors were tied to a promoter called CX3CR1, which is only active in microglia.
So the neurons didn't have the scissors, the liver cells didn't have them, only the brain's immune cells could cut the gene out.
Brilliant, right?
It is brilliant. But wait, earlier you mentioned that KAT7 is basically a fundamental tool for reading DNA. Don't the microglia need it to, survive early development?
They do.
Okay.
If you delete KT7 while the mouse is an embryo, it won't develop properly. So they added a third layer to the lock and key system.
The third layer.
Yeah, that's the ER part of the name. The microscopic scissors inside the microglia were designed to be completely inactive until they were exposed to a very specific drug tamoxifen.
I see where this is going.
Right. The researchers waited until the mice were fully grown adults already developing Alzheimer's plaques. Then they gave them the tamoxifen.
So they essentially flipped a switch that cleanly deleted the KAT7 librarian solely in the adult immune cells exactly when the Alzheimer's pathology was taking off.
Exactly.
What happened to the brain?
Without KT7 opening that specific page of DNA, the microglia had less CMPK2. That meant less excess mitochondrial DNA, which meant less leakage into the cytosol.
So the cGAS-STING chemical fire alarm was completely suppressed.
Completely. The mice had a massive, measurable reduction in their neuroinflammation. And amazingly, this led to a massive reduction in the actual amyloid-beta plaque burden in the brain.
Wait, really? The physical plaques went away?
They did. Because the microglia were no longer stuck in that hyper-paranoid loop of shooting chemical weapons at shadows, their behavior reverted to normal. They upregulated receptors like SCARA1.
Which are like their cellular garbage disposal receptors.
Exactly. They went back to their original jobs and started actively eating and clearing the plaques from the brain.
Okay, clearing plaques is amazing, but Alzheimer's is fundamentally a disease of memory. How do we know a mouse has its memory back? Like, how do you even measure cognition in a rodent?
They ran two major functional tests. First, electrophysiology. They looked directly at the synapses, the physical connections between neurons in the hippocampus at the Schaefer collateral CA1 synapses.
The hippocampus being the memory center of the brain.
Right. They measured something called long-term potentiation, or LTP.
Which is essentially how we measure synaptic plasticity, right? The ability of a connection to strengthen when we learn something new.
Precisely. In the standard Alzheimer's mice, that plasticity is practically destroyed. But in the knockout mice, that synaptic plasticity was beautifully restored.
But that's still on a cellular level. Did they run a behavioral test?
Yes, the classic Morse water maze. You place a mouse in a small pool of opaque water. Hidden just below the surface is a small platform.
And the mouse has to swim around, find the platform, and then use visual cues on the walls of the room to remember where it is for next time.
It's a pure test of spatial learning and memory. The standard Alzheimer's mice struggled terribly with this, but the knockout mice found the hidden platform significantly faster.
That's incredible.
They also did a probe test. They entirely removed the hidden platform and watched where the mice swam. The treated mice spent vastly more time purposely swimming in the exact target quadrant.
So they remembered where it should have been.
Yes, they proved their spatial learning and memory were rescued.
Let's just ground ourselves in what this means with our third recap.
Bullet 1. Scientists genetically deleted KT7 only in the microglia using the easy CRISPR method.
Bullet 2. This stopped the mtDNA leak and shut down the inflammation.
Bullet 3, the brain cleared the plaques, and synapse health and memory were fully restored.
Think about your own spatial memory right now. If you were dropped into a giant water maze, how well would you remember the hidden path? It really puts it into perspective.
Definitely does.
But here's the thing. Genetically engineering mouse brains is amazing for science, but we can't do that to human patients. We can't use a complex 3-part genetic engineering lock and key system in a neurology clinic.
No, we definitely can't.
So what does this all mean? How do we translate this into a real-world treatment? Is there a chemical compound, a drug that can simply bind to this KAT7 enzyme and block it?
There is. The researchers utilized a highly potent small molecule pharmacological inhibitor. It's called WM3835.
WM3835. So this drug essentially binds to KAT7 and prevents it from adding that negative acetyl charge to the DNA spools.
It confiscates the librarian's key so they can't open the book.
I love that. But how do you get a drug like that into the brain of a mouse to test it? I mean, the blood-brain barrier is notoriously difficult to cross.
For this initial proof of concept, they had to be very direct. They used an osmotic pump to deliver WM3835 directly into the fluid-filled ventricles of the brains of 5-month-old 5x FAD mice.
Wait, directly into the brain fluid?
Yeah, via intracerebroventricular, or ICV, injection, and they pumped it continuously for 4 solid weeks.
I have a massive concern here, though. If you pump a drug directly into the brain fluid, it doesn't just hit the microglia, it bathes every single cell in the brain.
True.
Doesn't blocking this fundamental epigenetic enzyme in our neurons cause brain damage?
It's a critical question, and the researchers anticipated it perfectly. Before they even tested the drug, they created a completely different genetic mouse model.
What did they do this time?
They deleted the KAT7 gene specifically in mature excitatory neurons, leaving the microglia alone. They wanted to see what would happen to an adult brain if neurons lost this enzyme.
And what happened?
Nothing. The adult neurons function perfectly. Their electrical currents, their synaptic plasticity, their overall health, all totally normal.
Oh, wow.
It turns out that while the brain desperately needs KAT7 during embryo development to grow, the mature adult brain tolerates its loss incredibly well.
That is a massive relief for the safety profile of the drug. So what were the results when they pumped the WM3835 inhibitor into the Alzheimer's mice?
The results basically mirrored the genetic knockout perfectly. The drug successfully lowered the H3K14 ACK marks across the brain. It reduced the clustering of paranoid microglia.
It cleared the plaques.
Cleared the plaques, and just like before, rescued the mice's memory in the water maze.
It proved that you don't need genetic engineering. You can achieve the exact same memory restoration with a chemical inhibitor.
Exactly.
But as incredible as mice are, many drugs cure Alzheimer's in mice and then completely fail in human trials. Did they test this on human biology?
They couldn't give the experimental drug to human patients directly, of course, so they took a brilliant alternative route. They used human-induced pluripotent stem cells, or iPSCs.
So for those who might not know, those are adult human cells, like skin or blood cells, that scientists chemically reverse engineer back into a blank slate stem cell state.
Exactly. They took these blank slate human stem cells and coaxed them into becoming human microglia-like cells, IMGs.
So they effectively had living human brain immune cells growing in a lab dish.
And then they intentionally exposed these lab-grown human cells to toxic amyloid-beta to trigger the panic.
And they pulled the alarm.
They pulled the cGAS-STING alarm immediately, churning out massive amounts of inflammatory cytokine IL-6. But when the researchers treated those inflamed human cells with the WM3835 drug,
the inflammation plummeted.
It stopped it dead. The drug blocked the epigenetic switch, and the human cells stopped releasing the toxic chemicals. It worked on human biology just as effectively as it did in the mouse models.
Let's do our final quick recap. Bullet 1, the drug WM3835 blocks KAT7 chemically. Bullet 2, when pumped into 80 mouse brains, it reverses pathology and memory loss.
And bullet 3, it successfully stops inflammation in lab-grown human brain cells.
Think about the magnitude of what that means. If science could offer you a single compound that pauses the aging and paranoia of your immune cells, how early in life would you want to start taking it?
It is a fascinating prospect. We are looking at the foundational blueprint for an entirely new class of medicine.
When you step back and look at the sheer density of the journey we just took, it's awe-inspiring. We traced a pathway starting from a single chemical acetyl group on a histone tail,
which upregulated a mitochondrial factory,
which caused a bacterial-like DNA leak,
which tripped a cGAS-STING alarm.
which destroyed the synapses required for memory. And we saw how blocking that very first step could restore memory in a living mammal. It really demystifies the disease. It's not just a vague cloud of aging, it is a highly specific mechanical failure.
And if we connect this to the bigger picture, modulating K-AT7 isn't just about Alzheimer's. Other studies have linked this exact same epigenetic switch directly to cellular senescence aging in other organs, like the liver.
So we aren't just talking about an Alzheimer's treatment. We're talking about a potential master switch for promoting resilience and longevity across the entire lifespan.
Exactly. By understanding how to carefully close these specific books of DNA, we might promote resilience against a wide variety of diseases.
It really makes you wonder about the very nature of how our bodies remember trauma. We started this deep dive imagining your brain as a mansion, with microglia as the guards.
Right.
Over time, those guards accumulate an epigenetic history, a physical molecular memory of every stress, every toxin, every microscopic injury they've fought.
And that history is what physically alters their DNA spools.
That history is what makes them paranoid and what ultimately causes them to pull the fire alarm and tear down the mansion. So I want to leave you with a final thought to mull over. If our immune cells' flawless epigenetic memories of past stress are what eventually caused them to destroy our brain, is the ultimate secret to a long-lasting mind simply teaching our immune cells how to forget.